Good afternoon, everyone. Welcome to class. Let's begin. I'd like to start with prayer, and so I would appreciate a volunteer. Please.
Dear Heavenly Father, we ask you to be with us today as we learn Dr. Gray. Please bless him that he'll be able to outline this material clearly, that we can understand and prepare for our exams. And we ask you to watch over all of us. So many of us are struggling, and that bless us with peace. And this we ask in the name of Jesus Christ.
Amen. Thank you. I apologize for last class. For those who are unaware, twice during the course of the hour, the sound system went out. Hopefully they've remedied that problem. Say they have, we'll see. I do have quiz number 4, so I'll distribute this. It is posted online as well. It will be due a week from today at 5 p.m. We should cover the material that I think already we've covered some of the material on the quiz, and I think we will have covered it all by the beginning of next week. So we'll go ahead and plan on it coming in Wednesday. If there's a delay, then I can push that back. But right now, let's shoot for Wednesday next week, 5 p.m. Okay, last class, we had begun talking about glycolysis. This is a very fundamental metabolic pathway involving 10 steps, 10 separate enzymes that convert glucose into 2 copies of pyruvate. Pyruvate is a molecule that is utilized further in the generation of ATP. And we will see that we will follow this process to the point that we actually look at the synthesis of ATP in the mitochondrial matrix. But for now, we are moving through glycolysis. I was asked if one should keep track of or know the electron pushing mechanisms for these 10 steps, and the answer is yes. 4 of them have a very similar mechanism. The kinases, steps 1, 3, 7, and 10, all carry out their reaction in a very similar fashion. But the others are more complicated, and we went through 2 of probably the most complicated reactions, and that's the hexophosphate isomerase, step number 2, and the aldolase reaction, step 4, in which fructose 1,6-bisphosphate is cleaved into 2 3-carbon pieces. The 2 products of the aldolase reaction are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. In the balance of glycolysis, only the glyceraldehyde 3-phosphate can be used. But rather than waste half of the potential energy from a copy of glucose, there is a 5th enzyme, a 5th step that we will talk about that is able to convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, allowing for both 3-carbon segments to move through the balance of glycolysis and produce pyruvate. All right, so that's sort of background for this. I am going to work at the board much of today, but information about the particular enzymes, some of the features of the active site, and even step-by-step depictions of the catalytic process are available on the slides. Some of this is also in the book. The book is not quite as thorough. There are also animations on Learning Suite and also embedded in the slides that go through each step in great detail. What I'm looking for is, in terms of electron pushing mechanisms, something comparable to what is seen on the slide. So that's sort of in between the book and the animations. If you want to look at the animations and learn those, that's great. Those will be just fine on the exam. But you can also look at the slides to prepare as well. So let's go back just for a second. Aldolase, this enzyme that catalyzes step 4 of glycolysis, actually occurs as a dimer, and the active site is an alpha-beta barrel. So you'll recall that there was a lysine that bound the fructose 1,6-bisphosphate, and then there was a glutamate or an aspartate, it's still debated, that initiated the reaction and brought about the cleavage of the fructose 1,6-bisphosphate. Okay, I'm going to pick up now with step 5, which is going to allow us to convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. The enzyme that catalyzes this is called triose phosphate isomerase. So it seems, at least on the surface, that this is a rather pedestrian function for this enzyme, and yet it is a remarkable enzyme. So there is, at the active site, a glutamate. So in fact, it is the particular glutamate that is going to participate in the reaction. And this has then a carboxylate with its negative charge. The substrate here is going to be our dihydroxy acetone phosphate. So the phosphate is here on the 3 carbon. Over here, there is a protonated base. It is assumed to be, in many models of this, it is assumed to be a histidine. So, right. So this process takes place also in an alpha-beta barrel. So the enzyme has folded spontaneously. It has dimerized, that is, there are 2 copies of this that are available, both of which are catalytically active. Both of them have this active site that contains this glutamate and this histidine or protonated base. All right. So this reaction is initiated by the extraction of a proton. This pair of electrons then comes down and generates a double bond. This pair of electrons comes out and picks up a proton. Okay, I'm going to go through this a little bit quickly. I apologize, but we'll never get through these reactions if I don't move along. Let me redraw this now with these changes in place. So we have this double bond. And we have now this rather symmetric intermediate. This is, again, is an enediol. Step number 2, the hexophosphate isomerase also utilized a enediol intermediate to interconvert glucose and fructose. So here we have this intermediate. Now, I forgot to tell you a very important feature of this enzyme. It has, in that alpha beta barrel, a flexible 10 amino acid flap. Okay, it is a piece of the protein that after the substrate binds will actually overlay the substrate. And it holds it in a planar orientation. That is to say that these hydroxyl groups and this phosphate are coplanar. As we generate this double bond, it's obviously, this region is going to be planar, yes. But that 10 amino acid flap, it moves about 7 angstroms to be able to overlay the substrate. It prevents rotation around this bond. If this phosphate became perpendicular, let's say to the board, it would lead to its spontaneous elimination, generating a toxic molecule called methyl glyoxal. Okay? So this flap then, this very convenient flap then holds this piece in place so that this structure is all coplanar and prevents the rotation of the phosphate 90 degrees with spontaneous elimination. Okay, so at this point we could go either way, right? This pair of electrons could abstract either of these, and that is the case, that this enzyme is reversible and it is driven by the substrate product ratio. So if we have high levels of dihydroxyacetone phosphate, then we are going to now remove this proton, and we're going to regenerate a carbonyl, but now on the 1 carbon. This is going to lead to this pair of electrons coming out and picking up this proton, and we now have glyceraldehyde 3-phosphate, which is the substrate for step number 6 in the glycolysis pathway. Okay? Questions about this? Yes?
You said they can extract, going backwards, they can extract either of those protons left?
It could, but if you have 100 times more of the dihydroxyacetone phosphate than the glyceraldehyde 3-phosphate, you will drive the reaction in the forward direction. So, but we will see with gluconeogenesis, this same enzyme actually produces the dihydroxyacetone phosphate from glyceraldehyde 3-phosphate. Okay, so this is step number 5. Step number 6 is the only oxidation step in the glycolysis pathway. The enzyme is called glyceraldehyde 3-phosphate dehydrogenase, and it involves as a second substrate NAD+. The NAD+ is going to be reduced, and the glyceraldehyde 3-phosphate is going to be oxidized to an acid. Okay. So let me sort of sketch out here very crudely the active site. So there is a critical cysteine present at the active site. There is also a site for NAD+, this second substrate. And there is a base that's going to abstract this proton. Again, it is not absolutely clear, but the assumption in many instances is that it is the histidine, oops, histidine. Now, okay, with a pair of electrons. So glyceraldehyde 3-phosphate, it exists as a tetramer, and each of these is catalytically active. Now, let's bring the substrate in. This is our glyceraldehyde 3-phosphate, and it is going to bind at the active site. This pair of electrons now is going to activate the sulfhydryl group of the cysteine, a little bit like our catalytic triad, okay? So remember, this has a much more generous pKa than did serine, but nevertheless, this is activated, so we remove this proton. This pair of electrons now comes and attacks this carbonyl, and we move the pi electrons out onto the oxygen nucleus. Okay, so this is step one of the process. Let me redraw this now with these changes in place. This is obviously a reactive species. It's not stable. We will reform the carbonyl. We'll regenerate that double bond. But now, rather than breaking this, the enzyme is able to direct the reaction in such a way that we transfer a hydride. Okay? So this now is a hydrogen with 2 electrons leaving. This constitutes oxidation. This is the actual oxidation step. The loss of 2 electrons for this species and the gain of 2 electrons on that hydrogen by the NAD plus to form NADH. Let me redraw this now. So the NADH, once formed, leaves, and we have, so this comes off, the NAD goes away, NADH, and we have this thioester. So formally now, this is not an aldehyde, but now a carboxyl group that has esterified to this sulfur. This thioester is a very high energy molecule. Put another way, we have activated this carbonyl, and it's going to allow us to couple a phosphate, which is not very reactive, to it. So far as we know, this phosphate comes from solution. The cell is filled with phosphate buffer. So here comes a phosphate in, and it is going to attack this carbonyl, will cleave the thioester bond, and we will re-extract this proton from the histidine. So in the end, we will have our histidine back in its original state. We will have this site open, and we will have coupled to our carbonyl here, a phosphate. So we've gone from a thioester to a phosphoester bond. So here we have now the 1,3-bisphosphoglycerate, which is the product of step 6. Okay, questions about this? Yes,
So what did you say is the name of the product?
it is 1,3-bisphosphoglycerate. Okay, so 1,3-bisphosphoglycerate. This is now glycerate, not glyceraldehyde. Okay. Step number 7 involves the formation. Yes.
So all of that, like the attacking of the cysteine and the formation of the phosphoester, is part of step 6.
Oh yeah.
And that's the only oxidation reduction reaction we have.
In glycolysis.
Yeah.
Yeah. When we get to the citric acid cycle, you'll have lots of oxidation reductions, but in glycolysis, this is the only step that represents a redox reaction. Okay. In step 7, this there is an enzyme, phosphoglycerate kinase, which is going to present an ADP. So let me simply ADP, and it's going to bind as the second substrate this 1,3-bisphosphoglycerate. The kinases, all of them have this sort of general structure where they have this sort of pie-shaped opening. The substrates are positioned next to one another at the apex of this opening. With substrate binding, there is closure. There's actually exclusion of water. The enzyme envelops the two substrates, and these are pushed right next to one another. In this step, then, the ADP, the phosphate here, is going to attack this one phosphate. This is now actually a reasonably good leaving group. And we are going to generate then ATP. So for one glucose, we end up with 2 glyceraldehyde-free phosphates. We end up with 2 copies of the 1,3-bisphosphoglycerate, and in step 7, we will generate 2 copies of ATP as our product, and we will leave then this 3-phosphoglycerate, okay? Okay, that's step 7. Questions? I apologize, I know I'm going through this quite quickly, not giving you much of a chance to copy things, but I do refer you back to the slides and the animations. Yes?
And then at the end of the step, this O- is stabilized by resonance. That's why it's still an O- just on the last carbon.
Oh, it's sufficiently acidic that the proton will come off in an aqueous environment.
But it's not reactive enough to do anything. It'll just sit there.
Yeah, it's not going to react at this point. All right, so 2 copies of ATP now. We're left with this particular product. Okay. Step 8 is an enzyme that is going to interconvert, or it's going to relocate the phosphate from the 3-hydroxyl group to the 2-hydroxyl group. Okay. So let me draw this out here. All right. At the active site, there are 2 histidines. So these histidines are positions close to one another and close to the substrate. One of these has a phosphate attached. So I'll just show that as follows. And the other has a pair of electrons. So it is not protonated. It has this pair of electrons. What happens then is that this histidine is going to remove this proton, and this pair of electrons is going to remove the phosphate. Okay. So this is actually a fairly odd arrangement. Having a phosphate attached to a histidine is not a common phenomenon. We do see it in biology, but it's not that common. And having a lone pair of electrons requires that that we manage the pH of the environment quite closely. So let me now redraw this to reflect the changes. So this is our intermediate. This is glyceraldehyde. This is 2,3-bisphosphoglycerate, BPG. So this is not the source of BPG that we talked about in the red blood cell that modulated oxygen binding. There is a separate system, enzymatic system in the red blood cell that generates the BPG, the 2,3-bisphosphoglycerate. Nevertheless, this active site is prepped by a copy of 2,3-bisphosphoglycerate coming in with transfer of a phosphate to one of the histidines to activate the enzyme so that it can process, this mutase can process the 3-phosphoglycerate coming from step number 7. Okay, so at this point, we now are going to bring this out. Well, it's a little differently. Come in with this pair of electrons. This pair of electrons now will come down and pick up this proton. And we will have then regenerated the active site to its original state, its catalytically active state, and we will have moved, in the end, we will have shifted the position of the remaining phosphate from the 3-carbon to the 2-carbon. So we now have this 2-phosphoglycerate. So phosphate back here, and we have this arrangement. So phosphate has gone from the 3-hydroxyl to the 2-hydroxyl, which prepares it now for step number 9 of glycolysis. This is the enolase step. Okay, let's move on. So enolase is interesting. It has 2 magnesiums at the active site. Now, some of these substrates with their phosphates will travel with magnesium. If you think about it, you kind of need to counterbalance the charge on the phosphate with some positive charge to be able to move these species into proteins, into an active site, because those tend to be, in many ways, surrounded by hydrophobic material. Okay, so here we have these magnesiums. At the binding site, we have the 2-phosphoglycerate, this, and we have All right. Okay. The enolase has a lysine that plays a critical role in this process, and I think there is a glutamate down here. As follows. Okay. The process, the enolase reaction, is initiated by this lysine, okay, with an amine with free electrons. Okay, it's not NH3 plus, it's NH2. So it's quite basic. This requires that we exclude water from the active site. But this will now come and remove this proton. So we abstract a proton, we generate a double bond here, and we push an electron out here on this oxygen. Notice now we have this carbon with two oxygens, both negatively charged, a very unstable, a very unlikely situation, except that we have these positively charged magnesiums to help stabilize this very reactive intermediate. So after this step, we end up with this. So we have some interactions here between these positively charged magnesiums and this species here where we've added additional charge to this carboxylate. Okay, this is not a stable situation, even with the magnesiums there. We're going to regenerate the double bond. We're now going to move this pair of electrons over here, and we're going to break off this hydroxyl group. It's going to come off and pick up a proton and leave as water. It's going to generate then a double bond. This is an example of a lyase reaction, where we eliminate water from a carbon-carbon bond and generate a double bond in its place. So let me go back to the product here. Final product. Here we eliminate water. We end up with this double bond. Okay. This is phosphoenol pyruvate. And in the final step of glycolysis, we're going to transfer this phosphate to ADP. Okay, so this is the pyruvate kinase reaction, step number 10. We're going to bring in ADP. We show it as follows. We're going to bring this in. We're going to move this here. And we are going to break this bond. It's going to come out and pick up a proton either from the enzyme or from the water environment inside the cell. But in the final step, step 10, the pyruvate kinase, we will generate 2 additional copies of ATP. So in terms of accounting for ATP, we had to use 1 copy as part of step 1 to phosphorylate glucose to glucose 6-phosphate. We had to use a 2nd copy in step 3 to add a phosphate to our fructose 6-phosphate to produce fructose 1,6-bisphosphate. But then in the sort of the latter part of glycolysis, we generate 2 ATP at step 7, and now we will generate an additional 2, 1 for each copy of glyceraldehyde 3-phosphate that went through the final steps in glycolysis. We'll generate 2 more ATP molecules. A net, for all of this glycolysis, a net of just 2 ATP. We also have 2 NADH molecules, which are energy rich as well and can be used as a source of energy for other reactions. Okay, so this walks us through then all 10 steps of glycolysis. The pyruvate actually is an interesting molecule. It is, even though it's transferring a phosphate to generate ATP, this step is actually energetically quite favorable. It is irreversible. And the reason for that is that the product, pyruvate, can undergo resonance stabilization through a process called tautomerization. Okay. I am now going to switch to slides. And take us right to formation of pyruvate. So, as mentioned, this final step is actually very energetically favorable because the product of this can undergo resonance stabilization. With the phosphate in place, it was unable to do that. But with the phosphate removed, we can now redistribute the electron density symmetrically over the oxygen carbon-carbon system that is shown here. So there's a keto form and enol form, and it can access both of these. And so the final step, even though we're generating ATP, which is energetically expensive, can be done because of the energy drop due to resonance stabilization here. The final step is irreversible in glycolysis. Yes?
So we talked about like a while ago how ATP likes to get rid of one phosphate to separate the charges or give the charges more room. Why doesn't the body just take it all the way down to just adenosine? Like why doesn't it get rid of 2 phosphates or 3 phosphates and just use that so you're not using as many ATP molecules, if that makes sense? Like you could just get rid of more phosphates.
I think the short answer to that is that it's not observed in nature. So is that a potential strategy if you were creating your own universe? Well, maybe so. What you do see is that sometimes you will see the removal of one phosphate and then you'll see another reaction that removes the second phosphate. Sometimes you'll remove the outer 2 phosphates as part of a single event. We will see that happening. But NAMP, I think, does have some energy to speak of, but mostly it's the ADP and ATP with their charge repulsion then that places them in a higher energetic state. And that leads to their being able to fuel, if you will, or provide energy for other reactions. I don't know that you could do that with AMP. There's no relief of charge repulsion because there's not 2 phosphates placed next to one another. Okay, let's talk a little bit about anaerobic metabolism. So our bodies exist and thrive in aerobic environments. Anaerobic conditions are generally need to be short-lived or they are very problematic and we can't survive very long in an entirely anaerobic condition. However, there are times, when you are unable to keep up with the oxygen demands of your body. So imagine a sprinter running 100 meters or a swimmer swimming 50 yards as fast as they can. Very often they breathe very little during that interval and consequently the tissues can be placed under somewhat anaerobic conditions. It's also possible that people with respiratory problems, such as chronic obstructive pulmonary disease, are unable to oxygenate adequately, and so they are constantly a little short on oxygen. Whatever the issue, when we don't have oxygen present, we're unable to carry out some of the processes that happen in the mitochondrium. Okay, we haven't even talked about the mitochondrium, but oxygen is reduced in the mitochondrial matrix, or at least in the membrane that divides the matrix from the intermembrane space. This process requires oxygen. If we don't have oxygen, we can't move electrons through the electron transport chain. We can't process pyruvate. We can't process NADH. And we're not generating any ATP in the mitochondria, and most of the ATP. So just as an example, glucose generates, one copy of glucose, if taken all the way from glucose to pyruvate, pyruvate to coenzyme to acetyl-CoA, and acetyl-CoA all the way through the citric acid cycle, producing high energy molecules that feed into the electron transport chain. If you have all of that in place, you'll generate about 32 copies of ATP. The net production from glycolysis is just 2. But if that's all that you have, if you don't no longer have a functional electron transport chain because of no oxygen, then this process is carried out. And the reason that this process of converting pyruvate to lactate is carried out is to regenerate NAD+. Okay? There is an absolute requirement for NAD+ at step 6 of glycolysis. If I don't have NAD+, glycolysis shuts down. So rather than having no ATP, the body then diverts the pyruvate and the NADH produced through this process, where we are going to reduce pyruvate to lactate, but oxidize the NADH to NAD+ to allow glycolysis to continue, given that it may be the sole source of ATP necessary for cell survival. So this is sort of a salvage, rescue kind of process that one doesn't want to do very often or for very long, but it allows cells to survive. Lactate buildup in skeletal muscle is painful. Your legs, if you run a marathon and you're really huffing and puffing at the end, it's usually an indication that you're under anaerobic conditions. You're trying to resupply that oxygen, but are not quite able. Under those conditions, you're going to be really stiff and sore for a few days. And part of that soreness has to do with the accumulation of lactate and its slow removal from peripheral tissues as you have normal metabolism taking place. Okay, questions? I think this is as far as I'm going to go today. All right, let me begin a story. I don't think I'll be able to finish it today. I think I mentioned that I had gone into the military, and at the end of that, I got out of the military too late to start graduate school that fall. The training lasted into the middle of the fall, and so I had to wait a year to start graduate school. And during the first part of that, I took a class or two, and then I then went to Germany. Actually, one of the classes that I took was sort of an upper division German class. I had actually been required to take German for chemistry. This was back when, this is a long time ago, but there was a time when the most significant chemistry journals were in German and not in English. And so chem undergrads needed to become reasonably proficient in reading scientific German. So I was taking an upper division sort of literature class, had nothing to do with science, but it was in German. And all of the other participants in the class were German majors, and they were about to go off to Germany. There was an arrangement between the university I attended and a university in Kiel, Germany, such that they took care of admission of these students into their program. They found them housing for the first term there, and then the students were allowed to choose which classes they wanted at the university in Kiel. And they all sort of encouraged me to come along, and so I went off to Germany. Well, first of all, I had to clear this with my National Guard unit, but they were very understanding, and it simply delayed my getting out of the military, but they gave me this, this opportunity to go to Germany to study. And so I went off with this group from my university to Kiel, where I was going to study Germany. Now, I tell you this story because during my time in Germany, I kind of did the things that the native Germans did, and did not do the things that I should have. And in the sense that I didn't go to church and I didn't keep all of the commandments. So I had this period of about 6 months. It was a very interesting, I learned a great deal, but I didn't grow spiritual, I'll have to admit. I came back from that and was just in time to go off to graduate school. I was going to go to graduate school in the eastern part of the U.S. And so I had just a couple weeks to pull my stuff together and pack my car and drive across the country to start grad school. But in that short interval, that couple weeks, I went to I just went back to my undergraduate campus, which was just in the same city where I live, and I happened to run into a young woman that I had known earlier. Her brother was a classmate of mine, same grade. Her family had belonged to a swimming club, and I had been a lifeguard at this swimming club, and would say hello to her. I mean, I would see her a few times during the summer, and would say hi. But anyway, she recognized me, I recognized her. We stopped and we started having this conversation. And she began to tell me about her life and sort of recent events. And it turns out that she was an extraordinarily bright person. She had garnered more hours of straight A than any other undergraduate. I think she was, not only summa cum laude, but was one of the valedictorians and so forth. She skied for the university. She was on their intercollegiate team. She was a member of the most popular sorority on campus. And she was fairly attractive too, which had nothing to do with my interest in her. So we'll leave it there. Thank you very much.